Hello! Now we will learn how to prepare a microscope slide of an onion skin and then examine its cell using an optical microscope. We will need the following tools: an eyedropper, a glass rod, tweezers, microscope slides, an onion, iodine solution, a microscope prepared for our joint work. Now, let's start the experiment. First, place a small drop of water on a microscope slide. Place a wet Mount Onion upper skin on the microscope slide. Place the cover slip on top of the wet Mount Onion skin. Now we set up the microscope and get an image of the cells. First, we observe a microscope slide of onion cell at low magnification. In individual cells, only the shells are visible. Nuclei are completely invisible. But pay attention. These inclusions over here are crystals. They can be mistaken for a nucleus. Now, let's adjust the microscope for high magnification. We turn on the lens, set the sharpness, brightness, and contrast. We now observe the nucleus, indicated by a red arrow, and thin strands of cytoplasm that penetrate the vacuole, indicated by black arrows. We begin to stain a microscope slide with iodine solution. We take a drop of iodine solution with a glass rod and apply it onto the microscope slide of onion skin, along with a cover slip. Now we wait until the microscope slide gets under the glass. Let's examine the stained microscope slide of onion skin at low magnification. We can see cells with different degrees of staining. Now we switch the microscope to high magnification. Take a look. The shell, marked with a red arrow, has become a dark brown color. The nucleus has turned yellowish brown. Plasmosmata, marked with a blue arrow, became stained and are now clearly visible. Vacuoles remained transparent and blue. Having conducted our interesting study of the onion skin microscope slide, we can now conclude that the nucleus, cell envelope, and plasmosmata are best observed under optical microscope. Crystals of large inclusions have been seen in individual cells.